now. Okay, so we're looking here at the syllabus. Today is the 3rd. We are going to definitely finish chapter 3 today. We'll make a good way through chapter 4. So we're still a tiny bit behind on the lecture portion of the class, but I'm not overly worried about that at all. You had this due today, but right at the start of class. I want to point out that I did post Monday the quiz for chapter 5, and the chapters have gotten longer. I think there's like 16 questions here, so it's a decent size quiz. I have changed it up a little bit since last semester to try and make, there was 3 or 4 questions which I deemed bad last semester, and I fixed those. And so I think the quiz should be slightly easier, to be perfectly honest, than it was last semester. Although I will point out the average grade on these Blackboard textbook quizzes is about 90%. So most, on average, people are getting A minuses on these quizzes, and that should still be the case with chapter 5. But well, this is when the chapters start getting a little bit harder, a little bit longer. And I will be posting this master and his emissary quiz later today. So another installment of that. And please do be looking out for the next video. I will be posting another 15-20 minute lecture video on the master and his emissary. My plan for one of the next 2 is to provide an extra credit opportunity in one of those. And it's an extra credit opportunity of my plan, which will also help you plan and strategize for your master and his emissary paper, which will be due on that last week of the semester. So be looking out for that. I will send out an email when I post it. Any questions or comments about assignments, things in general in the course? Cool. I'm not seeing much. So any questions? So we will go ahead and finish chapter 3, get started on chapter 4. Chapter 3, we've been talking about a whole bunch of different methods. Let's see, on slide 19. I do want to kind of just remind us of the different methods we've learned about. We started out talking about all of these different methods. The emphasis in this class is functional methods that compare brain activity between at least 2 different conditions, 2 different mental states, or 2 different behavioral tasks. That's what all these functional methods do. They measure brain activity from outside of the brain. And so they can all be used in humans because they are not invasive. Structural methods, on the other hand, people aren't performing a task. There isn't different conditions. You just basically an anatomical picture of the brain. Let's see. We talked a lot about EEG because we've kind of learned about 2 different kind of techniques. EEG is the general, as I mentioned last time, I spend a lot of time talking about EEG because if you are a neuroscience major, which the majority of people in this class are, a lot of people in this class are, you have to take 625. And the main method for measuring brain activity that we have access to in Lawrence campus is EEG. And we have 4 of them that could be used for the 625 class. So I spend a decent amount of time talking about EEG, more than the other methods, to help prepare you for that potential future class. And we talked about 2 different kind of approaches. EEG is just the general technique of placing electrodes on the scalp and then measuring electrical activity, which is coming from the brain with those electrodes. The first technique we talked about was frequency analysis, which is defined here, breaking that the waveform, the complex EEG, the electrical activity measured from at least 1 electrode, breaking it down into its different sinusoidal components. Sinusoidal just means kind of like a wave like that. These are the different sinusoidal components right here. Gamma, which is high frequency, beta, which is high but not as high as gamma, alpha, which we talked a lot about, which is kind of in the middle, theta, and then delta is the lowest frequency. The finding that I kind of emphasized, and is one that we actually replicate in 625, is that when your eyes are closed, alpha amplitude tends to be bigger, and when your eyes are open, alpha amplitude tends to be smaller. And that might reflect the fact that when your eyes are closed, you're kind of more restful. And when you're restful, the lower frequencies start to become more prominent in your electrical activity. I did show a video of that right here, where my eyes, I do the eyes closed, eyes opening. I'm not going to show it again, but that is a helpful video, I think. And this Fourier analysis, just another name for frequency analysis, and kind of the way of thinking about frequency analysis shown right here. This plus this plus this equals the raw EEG, or the complex EEG waveform, is what this green blue thing is supposed to be right here. Then last time we talked about event-related potentials, and in my opinion, this is when the EEG method starts getting a little bit difficult for people. We talked about frequency analysis. The other kind of technique that involves EEG is event-related potentials. There is no sinusoidal components. There is no frequency analysis. There is no Fourier analysis. You are just measuring kind of that raw EEG waveform between two different conditions. And you're always measuring it at some specific point in time. So we talked about the N170, which is measuring the responses of faces in this study, faces and cars at this specific time point, 170 milliseconds from the onset of those pictures. The pictures appear, either a face or car, 170 milliseconds. We're looking at the electrical activity, between the differences between those two conditions. And that is the N170 event-related potential. And you can see the difference in electrical activity right there. I also talked about the N400, which we're going to talk about more in the language chapter, which is another event-related potential, but it's measured 400 milliseconds after the onset of a word. And that word is a word that either satisfies an expectation, like pot in this example, or it violates an expectation, like fridge. And the N400 is the idea that there tends to be a bigger negativity in response to fridge, which is unexpected, compared to pot. There's a bigger negativity 400 milliseconds after the onset of the word fridge compared to 400 milliseconds after the onset of the word pot. And I showed another video which shows you exactly how ERP data collection works using the EEG stuff that we have at Fraser Hall. Moving along. MEG, I didn't talk about too much. We don't have access to one of those. They're expensive. They're expensive to buy, expensive to use. But I did mention these are kind of probably overall, I would say MEG is probably the best overall method because it has decent temporal and spatial precision. Last time someone asked, what is temporal precision? Temporal precision is the ability to see fluctuations in brain activity that are occurring over very small time scales, looking at the differences in brain activity between 1 millisecond and the next. That is high temporal precision. High spatial precision is being able to determine where in the brain those fluctuations are coming from. Fmri and PET, they both have good spatial but bad temporal resolution. That good spatial but bad temporal precision. EEG has very good temporal precision. That is, you can tell when things are happening with high precision, but EEG is bad at determining where in the brain that stuff, the brain activity is coming from. MEG is a good balance of all of the other methods. It has good temporal precision. That is, it can tell about the fluctuations that are occurring millisecond to millisecond in the brain, and it can tell where those fluctuations are coming from with pretty good spatial precision. The problem is, though, is that there's activity in the brain that, because of the shape of magnetic fields, cannot be measured by MEG. The activity that MEG can measure, it can do so with good temporal and spatial precision. But it's clear that there's some activity in the brain which is completely missed by MEG. Every one of these methods has a strength and a weakness. I'd say MEG overall has the best profile, but there's probably people that think Fmri, is the best method. If I had to choose one that I could use, MEG would be the one. But as I said, we do not have access to one in psychology department Fraser, at least to my knowledge. We start talking about single and multi-unit recordings. Now we're moving into methods which are used way less in humans. You only can use this type of a method on human if they're undergoing brain surgery for some medical reason. They're used a lot in animals. You're either sticking electrode deep into the brain, or you're at least sticking electrode onto the surface of the brain itself. These are way different than the other methods that we've talked about because these are clearly invasive. You have to open up the skull to get access to the brain, and you're sticking the electrode is literally touching the brain. It's either embedded deep into the brain, which someone asked, yes, that can injure the brain a little bit. In humans, the electrodes are always just placed on top of the brain, which presumably doesn't do much damage to the human brain. And I did show this, or I do have this video you can watch, which shows an example of single cell recording. Brain lesions are the best. Talked about this as well. Brain lesions are considered kind of like the best evidence in cognitive neuroscience. Of course, the problem with brain lesions, you can't randomly assign people to different lesion conditions. You can't say, okay, you're randomly assigned to have your left temporal lobe removed and you're the control condition. You can't do that. So the problem with brain lesion studies is that you can't really the random assignment is not something you can deal with. If you're using humans, there are plenty of studies where they'll take half the rats or half the animals and remove part of the brain or lesion part of their brain chemically. They're the experimental group, and then the other animals will be the control group. That happens all the time with animals, but with humans it can. And the brains are different enough between humans and animals that does kind of cause this chasm where we know a good deal about the brains of animals, but it's hard to know whether we can generalize what we know about animals to humans because there are enough differences that there's still questions that remain, and we can't study human brains as in depth and in detail as we can animal brains because ethics. Let's see. Yes, we'll talk more about this severing of the corpus callosum today. So here's another example of brain lesion research. So the severing of the white matter pathway that connects the left and right hemispheres. This is something which is talked about in Master and his Emissary. And we'll watch a video today. We don't watch lots of videos, but we will watch a video. It's important enough that I will share my sound, or I'm sharing my sound right now, and you will watch a video that is about the severing of the corpus callosum. It'll be at the beginning of chapter 4 on hemispheric asymmetry. TMS and direct stimulation. These are both methods which I mentioned you can't you can't randomly assign people to have a region of their brain damaged or lesioned. There kind of is an exception, which is TMS, which people were asking lots of questions about this last time. A TMS, you can kind of temporarily lesion a brain. I use the analogy of it's kind of like your hand falling asleep, where your hand is kind of numb for like a minute or two if you've been leaning on it for a while. And that's kind of what TMS does to specific regions of the brain. It can kind of put it to sleep for a minute or two. And then you can measure behavior before the TMS temporary lesion and then after the lesion during that segment of time when that part of the brain's asleep. You compare behavior between those two points in time, before and after the temporary lesion, which gives you an idea of what that brain region that you temporarily lesioned, what it does. And so these are, potentially could be a very useful method because you can randomly assign people to either have a lesion or not. It's temporary, but you can use random assignment here. I do already have a question. Let's take a look here. Can TMS make your hand and other parts twitch? Probably. I imagine if you use TMS on the motor cortex, which is this part, yes, Ben Murphy, one student is saying yes, it does. If you use it on the motor cortex, which is this part towards the back of the frontal lobe, I imagine that you could cause different parts of the body to actually move. Are there negative effects to different parts of the bodies with decreasing neural activity? Negative effects of different parts of the bodies and increasing. I imagine, are you saying that can TMS be dangerous? That sounds like the question you are asking. And I would say, yeah. I'm not exactly itching to be a participant in a TMS study. I don't know of a lot of evidence that it's dangerous. I imagine the brainstem, which is the lowest part of the brain right here, this controls vital functions. I wouldn't let anyone zap my brainstem, even temporarily, because 1 minute of the brainstem not working, I mean, the part of your brain that helps you breathe and your heartbeat, 1 minute of that being disrupted could obviously be not good. So I would definitely not just, on Friday night, I would not just, get a TMS zapper and play with it for fun or something like that. I would not. They're definitely a serious research tool, and I believe, they should only be used by professionals. And as I said, even it's a new enough technology that it makes me nervous that researchers don't necessarily know exactly how it works. It's assumed that the lesions are temporary. It just makes me a little nervous. There is definitely research and the participants seem to be okay. No, TMS is used for medical reasons too. In fact, I think TMS these days is actually used more for medical reasons than it is for research. I do know, I work in my graduate school, there was 2 researchers who did auditory stuff. They were kind of both my advisors. And shortly after I left, they were a husband and wife. The husband got access, he got a grant and then has access to a TMS now. Which is he's doing. So as right after I left is when he got the TMS, so I never got to use it or see how it works. But it definitely is used for research. I would imagine now that it's used more for medical reasons, it's particularly for treating medical conditions. Depression is the one that I've heard the most. That's the main medical condition which it's presumably been used for. And I don't know how, I don't think researchers really know how it supposedly works for depression. They just know that there's some evidence that using TMS can decrease the symptoms of depression. Moving on, yeah, you guys really like TMS. Every time we get there, the questions keep flowing, which is good. So moving on to direct stimulation. This is another method which you can cause, as a researcher, you can influence brain activity, but usually, once again, it's only used on animals or on humans who are having surgery. You can electrically stimulate different parts of the brain by basically taking a probe and on touching different parts of the brain, which can electrically stimulate the brain. And I think I mentioned last time that it was used to discover that motor homunculus, which is part of the frontal lobe. We talked about it in previous chapters. You stimulate different parts of the motor cortex, the motor homunculus, and you can cause different parts of the body to move. And so it suggests that there's kind of this representations of different parts of the body along the posterior or back part of the frontal lobe, which controls movements of the different body parts. Yeah. Okay, so we're now just a very quick review of everything that we did last time. So now I will talk about another method. This is another relatively new one, and this one involves genetic manipulations. So optogenetics, the opto reflects the involvement of light. Opto means some pertaining to light. And then genetics definitely means referring to genetics. So in optogenetics, through genetic manipulation, the researchers can change the nature of the neurons in at least parts of the brain. In kind of interesting but somewhat mysterious ways, they can, at least to me, the researchers can make it so that if a light is shined onto the neurons, that can cause those neurons to fire action potential. So that's the opto part. It makes the neurons light sensitive. Normally, neurons are in your skull and it's completely dark in there, but they use this genetic manipulation, which will make some neurons fire action potentials when a light is shined on them. And presumably some type of light is implanted into the brain, inside the skull. So the researcher, you can think of like a light switch, can turn the light on or turn the light off. And it's a light which is literally implanted inside the skull, adjacent to brain regions of interest. And one group of rats, the experimental rats, when the light is turned on, a certain set of neurons in their brain will fire action potentials. And then the control rats are just normal, when they will have a light surgically implanted into their skull, but their neurons do not fire action potentials when that light is turned on. They implant a light in both experimental and control rats to keep that as equal as possible between the two groups of rats. But only the experimental groups, when the light is turned on inside literally a light turned on inside their skull, inside their brain, that will cause certain groups of neurons to fire action potentials. And the researchers are interested. How does the behavior of the rat change depending on whether those neurons are firing action potentials or not? And here's just one example from the textbook. When those neurons were not firing action potentials. Well, so this is, I guess I should, a little background about rat behavior. If any of you have ever had a pet rat, during the daytime in particular, if rats are in like a square environment like this, they have a tendency to always be near walls, which is what you can see. And that's like the tendency. And here for this rat, the light is off. So there's a group of neurons. I don't even remember what region of the brain they were studying in the study. So I'm not going to ask you on a test, what region of the brain were they studying here? I want you to understand the general method of optogenetics. And this is an example of how they use this method to understand how changes in the brain activity can result in different behaviors. When those neurons are not firing action potential, the rat does what rats typically do during the daytime. They stay close to the wall. They're like wallflowers. And the hypothesized reason, which I think is pretty safe to say makes a whole lot of sense, is that this is kind of a way to keep themselves safe. they're not necessarily, during the daytime in particular, they're not going to be running across an environment because they feel exposed here, like as though predators can see them. And so during the daytime when it's light, they kind of like to hide as much as possible. So that's why they're wallflowers here. But then in the other condition, they turn on the light inside the rat's skull. That causes a subset of neurons in the brain to fire action potentials, and then they measure the behavior again. And you can see that here, the rat is now, his behavior has changed. These little lines represent where the rat is exploring, where the rat is walking around. And now the rat is much more comfortable walking through the middle parts. It's not worried about being exposed. Presumably, it's not as worried about being attacked by a predator. It's in kind of exploratory mode. And so this is a helpful example. Once again, I don't actually care about the specific brain region which is activated in the study. I don't remember what it was. I believe it was either the amygdala or the hippocampus, these middle temporal lobe structures, if I recall. What I do care about you understanding is that they're using optogenetic to turn off or on action potentials in some region of the brain, and then you compare the behavior of the animal when those action potentials are not firing and when the action potentials are firing. You compare, when we're dealing with functional methods, optogenetics would be considered kind of a functional method because there's always two conditions here: the brain, the light is off, action potentials are not fired. That's condition one. The other condition is the light is turned on and part of the brain is firing action potentials. Those are two conditions. And then we're comparing, in this case, behavior between those two conditions. And we're getting information about the function of that brain region that has been genetically modified to fire action potentials when light is shined on it. That's optogenetics. There is more about this in the book. Moving on. Functional connectivity analysis. This is definitely a more recent. Optogenetics is pretty recent. Functional connectivity analysis is also pretty recent. Although I haven't read tons and tons of studies with it, but I've read some review papers, I guess, about functional connectivity analysis. And I define it here as pairwise correlation between network nodes. So what I mean by network nodes, these are, I guess you could say, regions of interest in the brain. The brain has whole lots of regions in it, but you'll take certain regions that you're kind of interested in studying, and you say, here's a node. And we're going to use, I think probably the easiest way to example, you can use it with many different methods, but it's probably easiest to kind of think about it in terms of fMRI. And we'll be looking at brain activity among different parts of the brain. So we'll have the animal perform a task, for instance, or have a person perform a task. And then we're going to measure brain activity. I'll just pick, 3 nodes. We'll say the temporal lobe of the left hemisphere. The frontal lobe is the second node of the left hemisphere, and the parietal lobe of the left hemisphere. So that's 3 different nodes. A very simple experiment here with only 3 different parts: the temporal lobe, frontal lobe, and the parietal lobe, all of the left hemisphere. And we're measuring brain activity in each of these nodes. And for functional connectivity analysis, what the researchers are looking for is correlations in the amount of brain activity. So if we're using fMRI, that brain activity would be blood flow. But more generally, you can just call it brain activity for now. Pairwise correlations. And what this means is that there's kind of naturally a little bit of noise. There's a little bit of variability in the brain's response. So you could have an animal doing the same thing over and over again. And sometimes the brain activity in the temporal lobe, when it's doing that thing, will be bigger. And then sometimes doing that same activity, it'll be slightly smaller. So there's a little bit of noise. There's a little bit of variability. The rat does the same thing, but the brain's response while it's doing that thing is not identical every single time. There's a little bit of noise, a little bit of variability. What you're looking for in functional connectivity analysis is, if I'm pointing to my temporal lobe right here, on those trials where the brain activity in the temporal lobe increases, do we see a kind of similar increase in the frontal lobe as well? And when the activity on a given trial in the temporal lobe decreases, do we see that in the frontal lobe as well? And then we'd also do the same thing for the parietal lobe, which is back here. On those trials where activity in the temporal lobe increases, do we see kind of a similar increase and the parietal lobe as well. And on those trials where activity decreases in the temporal lobe, do we see a corresponding decrease in the parietal lobe as well? That's what is meant by pairwise correlations. You take these different regions of the brain and we ask the question on a trial by trial basis, is the amount of brain activity on each trial correlated, such that when one region of the brain has an increase in brain activity, the other region of the brain will have a corresponding increase in brain activity as well. The idea here is that you're searching for regions of the brain that kind of have long distance connections between them. The assumption, although it's sometimes true, sometimes it's not, that if two regions of the brain have correlated firing patterns, when this one fires a lot, this other one fires a lot too, there's a connection between them, so that one region of the brain is firing, it's sending a signal to the other region of the brain, which is causing it to fire as well. So this would be like synchronized or coordinated firing, which could cause these correlations. And so what you are aiming to find, you take a given node, like the temporal lobe, maybe that's the region you really care about, and you're looking for regions of the brain that are correlated with it. Some evidence suggests that the frontal and temporal lobes are highly connected with each other. And so you might expect on an experiment to find that there's high correlations between the frontal and temporal lobe. When the temporal lobe activation gets bigger on a given trial, the same thing kind of happens in the frontal lobe, but you might find that there's not a correlation between the frontal and parietal lobe. And that's what you're looking for. You're looking for regions of the brain that are correlated with a given region of interest and other regions of the brain that are not correlated. Those regions of the brain that show correlated firing patterns, you make the assumption, or it's oftentimes suggested, that they have some type of functional connection, that those two regions of the brain are working together. They're connected. And then finally is magnetic resonance spectroscopy. So my understanding, this is a very new method. I have not read a single paper about this, so I only know about it from the textbook. And in fact, I don't even think it was in the 4th edition. So this is a new addition to the 5th edition of the textbook for this course. It uses the technology of MRI and fMRI. So it uses the magnet, it uses those magnetic fields that are lined up when the magnet is turned on inside the brain, the oxygenated hemoglobin. And it's measuring neurotransmitters, or at least that's what it's trying to do. So this is interesting. Usually fMRI measures blood flow, metabolism, which is a proxy for brain activity. But this newer technology, magnetic resonance spectroscopy, is trying to actually look at the concentration of different chemicals in the human brain. This is pretty valuable because studying neurotransmitters in humans is notoriously difficult. In animals, I actually worked some on this as an undergraduate. The way you study neurotransmitters in animals is you literally stick a probe, you put a hole in their skull, literally with a drill, and then you'll stick a probe into that hole on their skull that then has a little bit of a little straw thing, which can kind of suck up some of the contents of the given part of the brain. And then you measure the concentration of different chemicals, neurotransmitters. That's how you do it in animals. Obviously, you can't do that on humans. Even if they're undergoing surgery, you can't stick a probe into their brain and suck out some of the neurotransmitters. And so this is an attempt at a not invasive way of studying neurotransmitters concentrations in humans. So that could be potentially very valuable. The problem is, as I said, I have not seen this a lot yet. I only heard about it in the textbook. The problem is, as far as I understand, is that you have to do a whole bunch of trials. So it's expensive. fMRIs are expensive to run. I think I said it's like thousands of dollars for an hour. So you have to do many, many different trials to get this technology to work. And the signals are weak. So it's expensive. And the findings are kind of, it's a very expensive method. And the findings are still not very strong. The effect sizes are small. Looks like we have some questions here. So the question is, how does that pass the IRB? How does that pass the IRB? Are you talking about the animal research where you stick?
Yes.
Actually, the animals have a different thing. It's not called the IRB. I forgot what it's called. If any of you guys are doing animal research in a lab, you might know what it's called. I did, and as an undergraduate, have to do something with that organization. I just don't remember what it's called. You could very easily Google it. But IRB is for humans only. And so there's a different set of people that review your applications if you're doing animal studies. And there's a whole different set of rules. Yeah, Aya Cook. Yeah, that does. Thank you. that does sound familiar. I believe that that's, I don't know what the acronym stands for, but that does it. That rings a bell. That sounds familiar. It's a different set of rules. And to be perfectly honest, the rules for animals are much, much, much more relaxed than they are for humans. And it makes some people uncomfortable. The guy that I actually worked with, I won't say his name, he was the professor. He's pretty famous. I didn't know it at the time, but then I graduated and realized that he was pretty famous professor. I did know that when I was there, the professor that I worked under, there were people that were animal rights people that would like protest in front of his house. So there are a lot of people that are uncomfortable with animal research, and they're not, and the rules are more relaxed. For instance, I mean, I actually quit because I was not personally comfortable with some of the things that they did in the animals in that lab, including things that I was asked to do. So I got into music perception research, which was much less invasive. But you know, with animal research, at least all the stuff they were doing in that lab, when the animals were done being experiments, they would kill them. And that was one of the things I didn't like. And that's just because the rules for yeah, you're welcome. Yeah, the rule, okay, International Animal Care and Use Committee. Thank you for looking that up. Yeah, I mean, someone just said last year, I don't know, I haven't verified the truth of this, but it says last year, organization killed monkeys after they're done testing. Yeah, it happens a lot. One of the main reasons they kill the animals is because, at least in the lab that I worked in, is that you want to make sure that the region, this is the main reason I was told, that you want to make sure that the region of the brain from which you were taking neurotransmitters or studying electrical activity was definitely the region of the brain that you were aiming for. And you can tell because you stick that probe into the brain, and then there's a little bit of damage from where the probe ended up. And then you kill the animal, and then you literally dissect its brain, and you look for the part of the brain where the probe was inserted. You can see the evidence of the insertion of the probe, and then you either know, yes, this was a successful experiment, we were measuring brain activity or neurotransmitters from the right area of the animal's brain, or we weren't. Yeah, so that's it. Let's move on.
I have another question. Sorry,
Yeah.
I think this is super fascinating. So, has there ever been like an experiment where, like, I don't know, like the psychology board was like, that was too much, like we can't do that again, like in the past with human stuff?
There was one study they did with, it was like an electroshock plate in a cage, and they basically like put the animal onto it and made it so that it couldn't run away. And then they had another test where the animal was able to jump off the shock plate from one side to the other. They were trying to show how you could get a learned helplessness. That one they said was too far.
Yeah, that was supposed to be like a model for depression, if I recall. Yeah, learned helplessness. I do remember that from as an undergraduate as well. I didn't realize that was actually a method that they ended up ruling could not be done anymore. But yeah, that definitely happens. But they let some pretty surprising stuff fly as well. When I was in graduate school, my friend was doing animal research, and she was telling me there are some researchers. She wasn't doing this, but there are researchers that actually look. I don't know if you guys were uncomfortable with the other stuff I was saying. I won't go into the details of this, but they study the neural correlates of pain in animals. And so the experimental group in that research would be animals which are inflicted pain. And they do it in different ways. And that's what I won't be specific about because I think it would be traumatic for some people. But that happens, and it is approved. The idea is if they will allow more uncomfortableness or pain to the animal to the extent that they think the findings will be, useful or helpful for humans. And so that's kind of the trade-off. All of these groups, they're reviewed. Humans are sitting in the IACUC groups and then the IRB groups, and they're having to make these decisions. Is the trade-off between what we'll learn, is it worth, the suffering that could possibly be caused? And with humans, the restrictions are much more relaxed than they are with humans.
Thanks for going into that.
Yeah. No, yeah. Thanks for bringing it up. So now we're moving into something which is much more, wholesome, which is the Stroop Task, which is not even really a neural method. Let's see what this chat is about. Yeah. Okay. So this student works in an animal care unit or an animal research lab, and the animal surgeries, which is that the animal surgeries are when you're like insert probes into their brain and stuff like that and do drills. And sometimes there are mistakes. I remember they had to train me how to do some of that stuff. And when you're first learning, it's hard. I mean, I'm drilling the hole into the skull of the animal. You had to like, I remember he told me, keep the drill touched against your cheek. So you have, you know, tons of control. You can like feel it, and you have to be careful because when you break through the skull with the drill, there's this tendency for it to dip down. And if you dip down too far, then you'll, you know, destroy the animal's brain. So there's a lot of, I mean, and this student saying that they're a little bit surprised by the lax rules. And I mean, I don't want to make a statement about that. Yeah, I mean, personally, I quit the lab because I was not down with it. Are there any other specific fields in neuroscience besides what you do that do not involve this kind of animal treatment? I know it's used quite a lot. That do not involve this kind of animal. Most neuroscience does not. I mean, only a subset. So I don't know the percentage, but most research in cognitive neuroscience does not do animal research. I'd say most of it doesn't. I mean, I do cognitive neuroscience research, and it's all in humans, and it's all none of it is invasive. Yeah. But yeah, so moving on to another task. This one is very wholesome, the Stroop task. And it's actually not even technically measuring brain activity, but it can be combined with other techniques to study the brain. And so I bring this up because this is kind of one of the quintessential cognitive tasks. And this course is cognitive neuroscience. So the cognition is kind of half of the course. And oftentimes what an approach to studies is that in this field, cognitive neuroscience, is taking a finding from this cognitive psychology and then adding a neural measurement. So they're doing the same thing that they did in just a pure behavioral cognitive study, but then you can measure the brain somehow, or you can look at the influence of different brain manipulations with lesions while people are performing a cognitive task. And so I'll show you this one, the Stroop task. It's also another task that we do talk about in, at least we did in the most recent semester of 625. It's a purely behavioral task, and we'll kind of go over it here. So there's 3 conditions which are shown here. And if you look at studies, studies sometimes use all 3 conditions. Usually when I do studies with this, I actually just use the first and last conditions here. This condition right here, excuse me, how embarrassing. This condition right here is kind of optional. I don't use it personally, but sometimes you'll see it in studies. So right here, this is a condition. If you look at the stimuli here, the color of the ink or the color of the letters is the same as what the word says. Red letters that say the word red, green letters that say the word green, blue letters that say the word blue, so on. And this is sometimes called the congruent condition because the color of the ink is the same as what the word says. This is like sometimes called the neutral or the control condition because the ink has colors, but it doesn't say anything. If you see, these are just all X's, so they don't say any words. So the letters have colors, the ink is colored, but the words don't say anything at all. That's neutral or control condition. And then finally, this is called the incongruent condition usually because the ink color is different than what the word says. So here we have red ink, red letters that spell the word green, green ink, which spells the word blue, blue ink that spells the word red. You get the point. Incongruent here. The color of the ink is different than what the word says. Question? Yeah. So the question is there much of a difference in results between the congruent and neutral tasks? Seems like they both be read quickly. So let me explain the way this task works, and then I will answer that question. I haven't really explained it. So one thing I want to point out is that the task that the participant performs in the study is not reading the words. That is not what the participant is asked to do. And I actually do want you to do, I was looking at the test 1. Last time I did this test for this class, one of the things I asked about this was, what is the tasks that people perform in the Stroop task? What are they asked to perform? The answer to that is they're asked to indicate the color of the ink. They are asked to indicate the color of the letters. They're actually, at least I actually run the Stroop task sometimes, and I actually specifically say, your job, your task is to indicate the color of the ink. Try to ignore what the word says when you read it. Don't read the word. And so it's easy, presumably, for the control condition, or the congruent condition rather, because you just say the color of the ink, red, green, red, blue, blue, green, blue, red. That's easy. It's also pretty easy for the neutral condition, once again, because you're just saying the color of the ink, green, blue, red, green, green, blue, red, blue. So that's pretty easy too. Things do get hard. So yeah, and someone asked, is there a difference? No, usually reaction times are about the same for these two. There might be slight differences in reaction time, but they're pretty slight. Usually the amount of time it takes to say red here is the same as the amount of time it would take to say red here. The congruent condition might be slightly faster, but it's not a big effect. However, a common effect is that people are slower when they're doing this. I'll kind of do it as a demonstration. I've done this a bunch, so I might be pretty good at it, but so you can kind of do it in your own head too. And most people get the feeling this is kind of hard, and it's a really consistent effect, really consistent. You can get significant differences between groups or between the congruent and incongruent conditions with like 20 participants, like every single time, very consistent. The difference is only like 100 milliseconds on average, but it's so consistent that it becomes statistically significant very fast. So red, green, blue, green, red, blue, red, green. So I did a pretty good job. Not probably quite as fast as I was here. But you know, if you look at it's kind of hard. Like the tendency, you want to look at the color of the ink, but the tendency is to say to read the word. It's hard to not read the word. So this is a task which is thought to represent attentional skills, your ability with attention, or your ability more specifically with executive function or inhibition. Your ability, especially in the incongruent condition, the thing you have to do is you have to inhibit that impulse to read the words. Our brains kind of want to automatically read the words because we had so much training reading the words, you know, back in the day, that we kind of want to automatically read the words. And so you have to use presumably your frontal lobe to put the brakes on that tendency, to inhibit it. And so you have to, I'm not going to read the word, and just look at the color of the ink only. So that's the cognitive thing, which is supposedly being measured here, your skills at inhibition. Looks like we have some questions. Yes, you administer this test, it's your job. And you have another condition where they switch back and forth between you reading the word and saying the color of the text. Interesting. I didn't know that there was ever a time where they read the word. That's interesting. Yeah, I'd use this as well, and I never have them read the word. And the studies that I've read tend to have them not read the word. I'd be curious to know what that accomplishes. We probably should, I mean, if you want to send that in a private message, you can tell me why you have them read the word. The other question is, so are people with ADHD consistently worse at this task? If there are any studies on that. You know, I haven't looked specifically at that. Theoretically, yes. Theoretically, they should be. This is supposed to measure attention. And so you would think that people that have a good, like a real diagnosis, some people are diagnosed with ADHD and they really don't have that bad of attentional skills, supposedly. I mean, I'm not a doctor, but I've heard of rumors like that. But people that have a legitimate diagnosis of ADHD, in theory, they should be fine. Here, I'll show you. People with ADHD should be fine in this condition because this condition doesn't really require attentional skills. It's easy. This condition, though, definitely does require attentional skills because you have to inhibit that impulse to read the words while you're just looking at while you're trying to cite the color of the ink. So yeah, people with ADHD should have impairment. They should be really, really slow. Everyone is slow on this, but people with ADHD should be extra slow in this condition right here. Looks like we have another quick question. Oh yes, okay, good. Thank you. I'll take a look at that later. Okay, so I'm not going to go into the details of this. I used to, but it's just too much. It takes too much time. I just wanted to show you how the Stroop task can be used to study, to get information about what parts of the brain are used or involved when people are performing the Stroop task. So this was a study where they looked at individuals who were lesion patients. So they had lesions in the left or right hemisphere. And then there was a control group that didn't have lesions at all. And then they had them perform the Stroop task. This is a long time ago. So instead of calling it the congruent, neutral, and incongruent condition, they called it the dots, non-color, and non-color words, and then color names. This is basically the incongruent condition. this is basically kind of like the congruent condition, and then this is like the neutral condition. Really, the key condition here is the incongruent condition. I'm not going to go into the details of the findings. Just know that this is basically this study, from 1974. So that's like 45 years ago or something like that. But they were looking at patients with lesions to different parts of the brain while they performed the Stroop task, and they were looking at patients with which type of lesion have struggle the most with the incongruent condition. You can see these are times right here, and you can see that there are definitely differences in the amount of time it takes to complete the task: 77, 43, 35, depending on where your lesion was. The control group had the fastest time, so the control group was least impaired in the incongruent condition, while the people with lesions had slower times, particularly the people with left hemisphere lesions. So you don't need to know the specific results of this study. I do want you to understand that this is a way that cognitive psychology studies are used to get information about the brain. So there is a quiz for this chapter, but I want to watch the video first. Chat. Yeah, okay, there's another. Yeah, that's still the private messages. Okay. So I do want to watch this video for chapter 4 real quick. I really want to get us started in this chapter. So because chapter 4, real quick, chapter 3 is now done. Chapter 4. This is on hemispherical symmetry. So this is a chapter which is important for, you're going to be writing this paper on the master and his emissary. The main topic of the paper is hemispheric asymmetry. Gazzaniga is an expert on hemispheric asymmetry. That's the main thing he studied. And this is the chapter in the book that he wrote about him. Chapter 4 is the chapter he wrote about hemispheric asymmetry. So you can there's a lot of connections that can be formed in this chapter compared to what you're reading about in the master and his emissary. There's lots of similarities, but there's also some differences. And I think I've shown you this exact slide before, but previously in the semester, we did not watch this video, but I now do want to watch this video right here. So hopefully this is still working. Did you guys hear that bell? Did you guys hear that? I want to make sure you can hear my sound.
Yeah.
Good, thanks. Okay, so we're going to watch this video. It's about 10 minutes. And then I'll give you more information about the quiz here in a second. Okay, so I'll go away for a second and start this video.
We began our journey into the human brain here on the campus of Dartmouth College in New Hampshire. I'd come to meet one of the world's leading brain scientists, Mike Gazzaniga, and a man he's worked with for over a decade. A man with 2 brains. You've been working a long time with Dr. Gazzaniga?
14 or 15 years.
It doesn't seem like that long, does it?
The collaboration began when Joe had surgery. And you had this procedure to correct an epileptic problem,
yes.
Is that right?
Trying to stop the seizures. I was having seizures like every day or so, or sometimes 2 or 3 a day.
To control Joe's epileptic seizures, a surgeon severed the connection between the 2 halves of his brain. Cutting the corpus callosum like this prevented the spread of the electric storms that caused his seizures. But it also prevented the left and right halves of his brain from communicating with each other. In the years since the operation, Joe's epilepsy has been under control. He now earns a living at an egg farm, and in his everyday life, he's largely unaffected by the fact that his left and his right brains work independently. Do you feel any different when you think about something than you did, any differently from the way you felt before the procedure?
It's just got a backup brain, that's all.
That's something everybody could use, right? I found out how true that was right away when I was asked to draw a different shape with each hand. In a brain like mine, roughly speaking normal, at least all in one piece, the left half of my brain controls the right side of my body, while the right brain controls the left side.
Oh, no.
But because the 2 halves are connected,
Nothing wrong with that.
getting each hand to work independently isn't easy.
Well, we're seeing the fact that Alan's hemispheres are connected, and that the motor messages from one are confusing the motor messages in the other,
I was just drawing an upside-down duck.
Okay.
But when Joe is given the same task, his two hands operate as if controlled by two separate brains. What's happening is that each half of Joe's brain is given a separate instruction. He's asked to fix his eyes on the cross in the center of the screen. Anything flashed to the right of the cross goes only to his left hemisphere. Things to the left go to his right hemisphere. Because the two don't communicate, each hand does only what its half of the brain sees.
Wow, look at that. It's really like two different people doing the same.
That's right.
Same
That's
task.
that's the idea. Okay, Joe, I want you to keep your hands out in front
In
of
an
you.
experiment that's now a classic in brain research,
Hemisphere.
Mike Gazzaniga over 30 years ago used a similar setup to find out if the two halves of the brain are specialized to do different things.
Ship.
Joe is being flashed a word only to one half of his brain. Words flashed to the right
Storm.
are seen only by his left brain, and Joe can report seeing those words just fine.
Piano.
Good.
But when a word is flashed to his right brain,
Didn't see that.
Okay. Joe, I'm going to ask you
but now watch what happens.
to draw that with your left hand.
You got me lost.
Why don't you try drawing another picture of it over here if that will help you.
Oh, phone.
Oh, okay.
Durr.
It's almost as though somebody has given him a secret communication.
That's
and
right.
now he knows that it's a telephone. But up until then, he was blind to it.
Exactly.
When Gazzaniga first did this experiment, it instantly proved that the ability to speak resides almost exclusively in the left hemisphere. Not until he sees what his right brain is drawing is Joe able to name
He
it.
said church, didn't he? After looking at the picture.
But he had to figure it out about as long as we did. That's really interesting. It's a picture here of somebody communicating almost with another person.
And the communication is not occurring inside the head, it's occurring out on the piece of paper.
Yeah.
Blob. I really don't know.
You want to draw a little bit more?
So far, Joe has been seeing only one word.
Cat. That's a cat
Things get
sound.
even stranger when he's flashed two words, each to only one half of his brain.
The right hemisphere saw toad. And
And so his left hand draws a toad.
So there's the toad.
Oh, it's a toad, right?
And this time I was able to guess what was coming.
Will they
Now-
put a little three-legged stool in there later, or what?
Joe's speaking left brain saw a stool. Saying the word lets the hand that's controlled by his right brain in on the secret.
That's great. That's really interesting. And if he had seen that with the corpus callosum intact, he would have drawn a toadstool.
Right.
Not a toad and a stool.
Right, exactly the point. I've been doing this for 35 years. Yeah.
And
It gets me every time.
Yeah, it must.
This time, instead of naming the word, I want you to point to the word.
Again, Joe sees two words simultaneously. Bell goes to his non-speaking right brain. Music to his speaking left brain. When asked to point to a picture of what he saw, he chooses Bell. But when asked why,
Yeah, why'd you pick that one?
Music.
Music?
and when asked to explain,
It was music, bell, and it was a few minutes ago. Last time I heard any music was coming from the bells out here,
Uh-huh.
banging away.
So the bell's
Yeah.
outside here.
So, look good enough answer to me.
what's extraordinary is that Joe's speaking left brain concocts a plausible story of why he pointed to Bell, even when some of the other pictures more obviously represent music. Gazzaniga believes this determination to find cause and effect, this desire to explain, is the left hemisphere's most marvelous property.
One of the unique things to the human brain is this need to interpret why two events occurred. What was the antecedent to this? What caused this? And if you can imagine that a species like us that has that little chip in its brain that asks those questions is going to survive rather well, because it's going to figure out more about the nature of the world than a species that doesn't have that.
Okay, Joe, I want you to keep your hands.
But as I was about to discover, the right brain has a very useful survival skill all its own.
What do you think will happen here?
So for you, we're doing a live experiment. Never done it before.
The experiment involves the 16th century Italian painter Arcimboldo, who made faces out of fruit, flowers, meat, even books. From other research, there's reason to believe that the ability to recognize faces is located exclusively in the right hemisphere. So Mike wondered if Arcimboldo's paintings would look different to each of Joe's two brains.
So will his left hemisphere say, I saw a potato, I didn't see a face? And will his right hemisphere say, I saw the face, and not comment on the fact it was made out of a potato?
You're going to see a figure followed by a choice of 2 words.
If this works, it'll be terrific, but we'll see. So here it is live.
Okay?
The first painting goes to the right hemisphere. And Joe points to
Okay, point
face.
it down.
The next painting goes to his left brain. And this time he points to fruits. Mike seemed pleased.
Are you having a moment?
This is too good.
Again to the right brain, and Joe sees it as a face.
Point it down.
But to the left brain,
Face made out of books.
Point it up.
Pointed to books.
Were you happy with what you're
Fruit.
doing?
It's unbelievable. He's doing it. You see that?
It's good. It's going so fast. It's shifting from left to right so fast. I can't keep up with you. You're used to looking at this.
When you show him a face in the right side, the left hemisphere,
Yeah,
he's focusing in on the elements that made up the face.
yeah.
When you show him the exact same picture in the left field, going to the right hemisphere, he's focusing on the face and not the elements.
And not the elements. If you came down from another planet and you saw faces and vegetables, you might not think there was much of a difference among them. But the brain seems to be made up in a certain way to say faces are very different from other objects.
That's right.
And one side of the brain specializes in faces. huh?
Exactly right. Exactly right. It is an adaptation that we have to detect upright faces. It's a very important. You can imagine in an evolutionary time that all of a sudden you have the ability to detect quickly an upright face. You want to read the expression on that face. You want to know if it's friend or foe. You want to have a set of questions about that face.
the right brain might be skilled at recognizing faces. But when it comes to what gives the human mind its power, the ability to reason, to invent, to interpret the world around it, Mike Gazaniga's 30 years of research has taught him which hemisphere he wouldn't want to be without.
The old phrase around our lab is, don't leave home without your left hemisphere. That's where the action is.
Let's see here. All right. So yeah, I just want to say like one little thing about that video. For one, we'll talk about this video next time. It explains a lot of, it goes over examples of some of the concepts that are in the textbook in this chapter, and we'll go into some of them in more detail. There is definitely some overlap between what was in that video and some of the things which are talked about in Master and His Emissary. For instance, the involvement of the right hemisphere in face recognition, the whole idea of the left hemisphere being involved as the interpreter that makes up stories about cause and effect. That's another thing, which is a big part of Master and His Emissary, actually. So I did want to say, though, that one of the reasons I showed that video was because of that quote at the end, don't leave home without your left hemisphere, which seems to go at, suggest that Gazaniga's opinion is that the left hemisphere is very important. It is very good, especially for a person whose job is a scientist like Gazaniga. Left hemisphere is important and he likes it a lot. And it's where the action is, the other quote. And I'm highlighting that because it's a definite difference to McGilchrist and the Master and His Emissary, who has almost the complete opposite perspective, that the left hemisphere is in some ways evil and kind of dishonest, and that the right hemisphere is more down to earth in a sense. And so it kind of shows, and this is one of the few things in the course which is like directly relevant to the Master and His Emissary paper. There's definitely some differences between the two authors, although there's some similarities as well. So we will talk about that video more next time, but it's been a pretty long class today, so we'll call it a day, and I will see you Monday. Look out for a new Master and his Emissary video coming up in the next couple days. I should have time to record it Thursday or Friday.
Thank you.
Thank you. See you later.